Hello, everyone. My name is Stephanie Hernandez, and today I will be giving a small presentation about the cell cycle regulation in the cell. I hope everybody enjoys it and we all learn something new today. Here's my first slide. It's a small introduction. It's called Cell Cycle Regulation, Ensuring Proper Cell Division. Number one, ensuring proper cell division. Cell cycle regulation ensures that cells divide at the right time and maintain proper size and function. This regulation is crucial for the accurate distribution of chromosomes to daughter cells, preventing aneuploidy and genomic instability. Number two, growth and development. During organismal growth, precise control of the cell cycle ensures that tissues and organs develop correctly to the appropriate size. In embryonic development, tightly regulated cell cycles are necessary for the orderly progression of developmental stages and differentiation of various cell types. Number three, we have tissue maintenance and repair. Adult tissues rely on regulated cell divisions to replace cells that are lost due to injury, apoptosis, or neutral turnover. Stem cells and progenitor cells must divide in a controlled manner to replenish tissues without leading to overgrowth or depletion of stem cell pools. Number four, prevention of uncontrolled cell proliferation. Proteins like p53 in retinoblastoma (RB) act as tumor suppressors by halting the cell cycle in response to DNA damage or other cellular stressors, allowing for repair or triggering apoptosis if necessary, if damage is irreparable. A small summary about this slide will be that cell cycle regulation is vital for the controlled growth, development, and maintenance of healthy tissues. It ensures orderly cell division, preserves genomic integrity, prevents tumor regenesis, and allows tissues to respond adaptively to physiological needs and external stimuli. Our next slide here is called the cell cycle overview. We have G1 phase, which is also called GAP1. Description. The first gap phase after cell division. Cells grow in size, produce RNA, and synthesize proteins necessary for DNA replication. The duration of this is variable, depending on cell type and conditions. Next, we have the S phase, which is also called synthesis. The description of this, the phase where DNA replication occurs. Each chromosome is duplicated to produce sister chromatids. The duration of this is typically in between 6 to 8 hours in mammalian cells. Next, we have G2 phase, which is also called GAP2. The description of this, the second gap phase after DNA replication. The duration of this is typically in before 4 to 6 hours in mammalian cells. And last but not least, we have the M phase, which is mitosis. The description of this, the phase where cells divide occurs. It consists of mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. The duration of this is typically in between 1 to 2 hours in mammalian cells. A summarization of this slide will be that each phase of the cell cycle is tightly regulated to ensure accurate DNA replication and division, maintaining cellular function and genetic stability. Disruptions in this regulation can lead to uncontrolled cell proliferation and obviously cancer. Excuse me. The next slide here is called regulation at G1 checkpoint. The G1 checkpoint is also known as the G1/S checkpoint or restriction point. In mammalian cells is a critical regulatory juncture in the cell cycle. The primary purpose of the G1 checkpoint is to ensure that the cell is ready to replicate its DNA and divide. Purpose and importance. Assessment of the cell size. The cell must be large enough to ensure that it can support two daughter cells after division, excuse me. Nutrient availability. The cell must have sufficient nutrients and energy reserved to support DNA synthesis and cell division. Next, we have DNA integrity. The cell checks for any DNA damage. If damage is detected, the cell will either repair the damage, or if the damage is too severe, it initiates apoptosis, which is programmed cell death, to prevent the propagation of mutations. That's what happens to prevent, that's what it prevents the propagation of mutations. Next, we have extracellular signals. The cell responds to external growth signals, such as growth factors. In the absence of these signals, the cell might enter a quiescent state, also known as the GO phase, instead of continuing through the cell cycle. Some of the outcomes of G1 checkpoint activations are progression to S phase, cell cycle arrest, and apoptosis. The G1 checkpoint is a vital control mechanism that ensures cells only proceed to DNA replication and division under optimal conditions. It maintains genomic stability and prevents the propagation of damaged or inadequate cells, playing a crucial role in normal cellular function and preventing diseases such as cancer, which I mentioned before. Here is the next phase, which is called regulation at S phase. Ensuring accurate DNA replication is crucial for maintaining genetic integrity and preventing mutations that could lead to diseases such as cancer. Some of these mechanisms are: number one, DNA polymerase accuracy, proofreading activity. DNA polymerases possesses an intrinsic proofreading ability. As the enzyme synthesizes a new strand, it checks each nucleotide it adds. If an incorrect nucleotide is incorporated, the polymerase detects the mismatch, pauses, and excises the incorrect nucleotide through its 3 to 5 exonuclease activity before resuming synthesis. Number two, mismatch repair system. Post-replicative correction. Even with proofreading, some errors may escape correction during synthesis. This mismatch repair MMR system corrects these errors in replication. Proteins such as MUTS and MUTL in prokaryotes, actually MSH and MLH in eukaryotes, recognize and bind to mismatch bases. And number three, origin of replication in licensing. Precise initiation DNA replication begins at specific sites called origins of replication. These sites are tightly regulated to ensure that replication starts only once per cell cycle. That's very important. In eukaryotes, a protein complex known as the origin recognition complex, which is also called ORC, binds to replication origins. This is followed by the recruitment of additional factors, such as CDC6, CDT1, and MCM helicase, forming a pre-replication complex during the G1 phase. By integrating these mechanisms, cells achieve highly accurate DNA replication, minimizing the risk of mutation and maintaining genetic stability across generations. Here we will talk about regulation at G2 point, also known as G2/M checkpoint. It's a crucial regulatory point in the cell cycle that occurs at the end of that G2 phase, just before the cell enters mitosis, which is also, as we already learned, the M phase. The primary purpose of the G2 checkpoint is to ensure that cells do not initiate mitosis until all DNA has been accurately replicated and any DNA damage has been repaired. Purpose of importance of this: verification of DNA replication. The G2 checkpoint ensures that all of the DNA has been completely and accurately replicated. DNA damage repair. The checkpoint checks for DNA damage that might have occurred during replication. It provides time for the cell to repair this damage before entering mitosis, thereby maintaining genetic stability. There are some key components and mechanisms such as sensing DNA damage and replication, status regulation of cell cycle, progression, p53 pathway, DNA repair mechanisms. The G2 checkpoint is a critical control mechanism that ensures cells do not enter mitosis with damaged or incompletely replicated DNA. It maintains genomic instability by allowing time for DNA repair and preventing the propagation of errors, playing a vital role in normal cell function and preventing diseases just such as cancer. Next, here we have regulation at M phase, which is also called the spindle checkpoint. The spindle assembly checkpoint, also known as SAC, and is also known as the mitotic checkpoint, is a critical regulatory mechanism that ensures proper chromosome segregation during mitosis. Purpose and importance: ensuring proper chromosome attachment. The SAC ensures that all chromosomes are correctly attached to the spindle. Microtubules via their kinocores before the cell proceeds to anaphase, preventing chromosomal instability. By halting cell cycle progression until proper attachment is achieved, the SAC prevents chromosome missegregation, which can lead to aneuploidy, an abnormal number of chromosomes, and genomic instability, often associated with cancer. Mechanisms of the spindle assembly checkpoint. Number one, sensing incorrect attachments, kinocores. Kinotores. The kinetochore is a complex assemble at the centromere of each chromosome. Then we have attached kinochromes and unattached or improperly attached kinetochromes. Generate a weight signal that activates the SAC. The spindle assembly checkpoint is a vital control mechanism that ensures accurate chromosomes segregation during mitosis. Next is role of tumor suppressors in cell cycle regulation. P53 tumor suppressor protein. P53, often referred to as the guardian of the genome, is a crucial tumor suppressor protein that plays a vital role in maintaining cellular integrity by regulating the cell cycle and preventing the proliferation of damaged cells. Structure and function. Gene encoding P53. The TP53 gene encodes the P53 protein. Mutations in this gene are found in more than half of all human cancers. So, which means that having a correct mutation of this P53 is very important to not develop cancer. Retinoblastoma, RB, tumor suppressor protein. RB, encoded by the RB1 gene, is another essential tumor suppressor protein that regulates the cell cycle and ensures proper cell proliferation. The structure and function of this gene encoding RB. The RB1 gene encodes the retinoblastoma protein. Mutations in RB1 are associated with retinoblastoma, which is a rare childhood cancer and other malignancies. In conclusion, here for this slide, P53 and RB are crucial tumor suppressors proteins that regulate the cell cycle and ensure genomic stability. P53 responds to cellular stress by inducing cell cycle arrest, apoptosis, or DNA repair, while PRV controls the G1/S transition by inhibiting E2F transcription factors. Next slide is oncogenes and cell cycle dysregulation. Oncogenes are genes that have the potential to cause cancer. In their normal state, they are called proto-oncogenes and play crucial roles in regulating cell growth, differentiation, and survival. However, these genes become overexpressed, they can drive the uncontrolled cell proliferation characteristic of cancer. Nature and function of oncogenes. Proto-oncogenes versus oncogenes. Proto-oncogenes. These are normal genes that encode proteins involved in promoting cell growth and division, such as growth factor receptors, signaling molecules, and transcription factors. Now we have oncogenes. When proto-oncogenes undergo mutations, become amplified, or are placed under the control of strong promoters, they can become oncogenes. Mechanisms of activation, mutation, gene amplification, chromosomal translocation. Key oncogenes. We have MYC and RAS. Oncogenes like MYC and RAS and RAS play critical role in cell growth and survival, and their dysregulation can lead to cancer. In this slide, we will talk about cell cycle regulation in cancer. Cell cycle dysregulation is a hallmark of cancer as it leads to uncontrolled cell proliferation and the accumulation of genetic abnormalities. The cell cycle is a tightly regulated process that ensures cell divide only when necessary and that any DNA damage is repaired before division. When these regulatory mechanisms fail, it can result in cancer, as I've mentioned before, of course. Specific failures in cell cycle regulations are linked to various types of cancer. I have here a few examples. Number one, ovarian cancer. Ovarian cancer is the mutations of the BRCA1/2 And the TP53 that we had talked about, when these have mutations, it can occur, it can end up in ovarian cancer. Number two will be retinoblastoma, which is the mutation of the loss of the RB1 gene, we also talked about before. Next, which is a very type of common cancer, is breast cancer, which is the overexpression of cyclin D1 and a mutation of the TP53. See how this TP53 keeps coming up in various types of cancer. That's why it's so important, the healthy replication of this TP53. Next, we have colorectal cancer, which is a mutation in the APC gene and the KRAS gene. The KRAS gene we also talked about before. Here we're going to see that gene again in lung cancer, which is a mutation of two of the genes that we talked about already, the KRAS and the TP53. Here is a type of cancer that I've seen a lot in my medical career, which is pancreatic cancer. Pancreatic cancer is a mutation of the KRAS and inactivation of the CDKN2A. Next, we have glioblastoma, which is a mutation in the TP53, amplification of the CDK4, and a loss of the CDK2N. See how melanoma and glioblastoma have the same gene mutation, causes the same different type of cancers. Same mutation with different type of cancers. Here next we have, and last we have, a chronic myeloid leukemia, also known as CML. This is a mutation of the BCR ABL fusion gene. When these two genes fusion is when this type of leukemia happens. Here we will talk about the therapeutic targets in the cell cycle regulation. Targeting cell cycle regulators have become a promising strategy in cancer therapy, as this regulation of the cell cycle is a hallmark of cancer. Various therapies have been developed to specifically inhibit the key proteins and pathways involved in cell cycle progression. Here we gotta talk about some significant approaches. Number one, CDK inhibitors. These inhibitors specifically target CDK4 and CDK6, which are involved in the G1 to S phase transition by phosphorylating the retinoblastoma protein. See how important these inhibitors are. Number two, we have checkpoint kinase inhibitors. Inhibitors of CHK1 impairs the G2/M checkpoint, forcing cells with damaged DNA to enter mitosis, leading to cell death. Number three, here we have aurora kinase inhibitors. Inhibition of aurora kinases disrupts mitotic spindle formation and cytokinesis, leading to cell cycle arrest and apoptosis. Here, number four, we have WEE1 inhibitors. Inhibition of WEE1 prevents the inactivation of CDK1, leading to premature entry into mitosis and cell death, especially in cells with TP53 mutations. MDM2 inhibitors. MDM2 inhibitors prevent the interaction between MDM2 and P53, leading to P53 stabilization and activation of its tumor suppressor functions. In summary, targeting cell cycle regulators offers a strategic approach to cancer therapy. By exploiting cancer cells, these regulated cells cycle machinery. We have gotten almost to the end of this presentation. So here is my last slide, a brief conclusion of everything we just learned. Importance of cell cycle regulation. Ensures orderly and timely progression through the phases of the cell cycle. G1, S, G2, and M, also known as mitosis. Phases of the cell cycle. We have G1 phase, cell grows and prepares for DNA synthesis. Critical regulatory point at the G1/S checkpoint, S phase, DNA replication occurs, ensuring each daughter cell receives a complete set of chromosomes. G2 phase, cell continues to grow and prepare for mitosis. It includes the G2/M checkpoint to verify DNA replication is complete and undamaged. End phase, mitosis. Division of the cell nucleus followed by cytokinesis resulting in two daughter cells. Checkpoints in the cell cycle. As we said before, G1/S and G2/M. In the spindle assembly is a key regulatory proteins. We have cyclins and CDKs, which we talked about, tumor suppressors, and CDK inhibitors. We already talked about how important those were. In conclusion, proper regulation of the cell cycle is crucial for maintaining cellular integrity and preventing uncontrolled cell proliferation. Dysregulation of the cell cycle can lead to cancer, and understanding these mechanisms provides opportunities for targeted cancer therapies. Here is the slide list of references where all the information that we already talked about can be found. Thank you, everybody, for paying attention, for listening. I hope everybody liked it, and I also hope everybody have a good day. Thank you.